Bismillahirrahmanirrahim. Respected teachers and my dear fellows, Assalamu alaikum. Welcome to our today presentation. The topic of my presentation is Architecture of Bacterial Cells, which is presented by Saira Tabassum, MPhil second semester, Department of Microbiology, MB3 2022-2012. The course instructor of my subject is Dr. Niaz Muhammad, Chairman of MLT Department Kohat and Kohat University of Science and Technology, Kohat. The contents of my presentation includes introduction, eukaryotes versus prokaryotes, classification of bacteria, structure of bacteria, conclusion, and at the end references. Now, what is cells? Cells are the smallest unit. A cell is a structural, functional, and biological unit of an organism. Cells are the basic building block of our muscles, bones, and other organs. All living things are made up of cells. The most cells are microscopic. Now, the persons who first to view cells in 1665, Robert Hooke used a microscope to examine a thin slice of cork. What he saw looked like small boxes, so he called them cells. Now, what is the cell size? The dimensions of most bacterial cells are its diameter is 0.2 to 2.0 millimeter. Human red blood cells is about 7.5 to 10 millimeter in diameter, and its length is 2 to 8 millimeter. Some cyanobacteria are made up of 60 millimeter long. Bacterial cells have large surface to volume ratio. Therefore, all parts of the cells or close to the surface can be quickly reached by nutrients. Now, cell types. There are two different types of cells, prokaryotes and eukaryotes. Distinguished features of prokaryotic cells. DNA is not enclosed within a nuclear membrane. A single circular chromosomes. Not associated with histone proteins. Lacked membrane enclosed organelles like mitochondria, chloroplast, Golgi apparatus, etc. Cells walls have usually contain peptidoglycan, a complex polysaccharides unit divided by binary fission. Now, these are the main differences of prokaryotic and eukaryotic cells. On the right hand, eukaryotes have a nucleus, also have organelles, have a cytoplasm, and it can be multicellular or unicellular. But on the other side was prokaryotes have no nucleus, small and simple, no organelles, very abundant. It's a unicellular and all are bacteria. But in between there are some similarities of prokaryotic cells and eukaryotic cells, like they both have ribosomes, both have DNA, and they both have cytoplasm, have a cell membrane, and have a flagella. Now, the shapes of the prokaryotic bacteria, some are cocci shaped, like it means spherical, some are rod-like, bacilli, and some are spiral. Bacteria. What is bacteria? Microscopic unicellular organisms belong to kingdom Monera. that possesses a prokaryotic cell structure. They reproduce by binary fissions and by forming spores. They can inhabit all kinds of environment such as in soil, acidic, hot springs, radioactive waste, seawater, deep in the earth crust, and stratosphere. The microscopic studies have revealed that the ultrastructure of bacterial cells possesses the following structure, like capsule or slime layer, cell wall, and its have a plasma membrane, like flagella, fimbriae, and pellets. The cytoplasms, which have include ribosomes, mesosomes, fat vacuoles, inclusion bodies, and nucleosides. What is capsule? Certain bacteria like Diplococcus and pneumococcal are extremely covered by a layer, and that layer is called capsule. What is glycocalyx? Polysaccharides on external surface forms an envelope around the cell, and it can be observed under light microscope. Chemically, capsules are made up of proteins, polysaccharides, and lipids. It's a sticky in nature, secreted by cell wall, which gets firmly attached to cell wall. Many have gram-positive bacteria and gram-negative bacteria. A regular structural layers called S layer on their surfaces. Now, what is cell wall? A typical bacteria cells possesses a rigid structures called cell wall that lies mostly outside the plasma membrane. It's about 10 to 20 nanometer in thick and 20 to 30% of dry weight of the cell. It's responsible for maintaining the shape of the bacteria, and it prevents the cell from osmotic lysis, means mostly for breakdown. The chemical substance that confer rigidity to the cell wall is peptidoglycan. It's a most strong polymer found in the prokaryotes. Now, bacteria are classified into gram-negative and gram-positive. It depends upon the nature of the cell wall. On the right side, the gram-negative bacteria show pink colors under observed in the microscopy, and on the other hand, gram-negative positive bacteria show purple color. Now, these are the main and big differences between the gram-negative and the gram-positive bacteria. Now, in the gram-positive bacteria, the peptidoglycan layer is thick, but in gram-negative, it is thin. Teichoic acids are present in gram-positive bacteria, but it is absent in gram-negative bacteria. And periplasmic space is also absent in gram-positive bacteria, but it is present in gram-negative bacteria. So all these different features have included both gram-positive and gram-negative. Now, in the next slide, what is plasma membrane? The most widely accepted current model for the membrane structure is fluid mosaic model of Singer and Nicholson. According to the fluid mosaic model, it's a lipid bilayer consists of two types of protein, peripheral protein and integral protein. Now, first we define the peripheral protein. They are loosely connected to the membrane and can be easily removed and make 20 to 30% of total membrane proteins. Integral protein, 70 to 80% of membrane proteins are called integral proteins. They are not easily extracted from membrane. These are the dramatically structures of the plasma membranes, which includes on the right side glycoproteins, hydrophobic, hydrophilic, but on the other hands in the left sides it's glycolipid, phospholipids and integral protein. What is pili or fimbriae? Many gram-positive bacteria have short hair-like appendages rather than flagella and not involved in motility. They are usually called fimbriae. Although a cell may be covered by thousands of fimbriae, they are visible only under electron microscope. They seem to be slender, like helical arranged proteins, subunits, and are about 3 to 10 nanometer in diameter and up to several micrometer long. At least some types of fimbriae attach bacteria to solid surfaces such as rocks and streams and hard host tissue. Flagella. About half of all non-bacteria are motile. Most of them are use flagella. They have long, thin, helical appendages. A bacterium may have one or several flagella, which can be in the following arrangements, like monotrichous, single polar flagellum at one end, amphitrichous, two polar flagella, one at each end, lymphotrichous, two or more flagella at one or both ends, peritrichous, many flagella over entire cell surface. Now, these are different arrangements of bacterial flagella. And at the end, conclusion. Many bacteria have external structures that increase their survival and pathogenicity. DNA is not enclosed within a membrane in its one circular chromosomes. Their DNA is free, not attached to proteins. They lack membrane-bound organelles. Also, their cell wall is made up of peptidoglycan. It's divided by binary fusions, and they have a very small in size. I mean, the cell is very small in size. And these are my presentation references. Thank you.